genetic basis of antibody structure. So far, I've been talking about diversity, specificity, and if you remember me saying that the antibodies and the B cells and the T cells have the capability to recognize antigen. But then you wonder, there are millions of antigens there. How would our body respond to them in a very specific and precise way? What's the mechanism there? So that mechanism is coming from the genes, the genetic basis for that, and that's what I'm going to discuss today. And keep in mind, I gave you a simple analogy, the 26 letters, but all the volumes in the world, the newspaper, everything daily people speak, there's always new thing coming up with pretty much the same thing in terms of the alphabets. So likewise, in our genes, that's what we need to know. How would those factors play out in terms of our recognition of a foreign antigen? So our bodies have a capability that may not kind of perform to the fullest, but they have this capability of seeing maybe 10 to the 11 different type of antigens. So you can imagine enormous capability. So anyway, today I'm going to talk about some of the structures, especially how the genes are expressed in antibody, some of the events related to genetics. As I said the other day, I'm going to talk about class switching. If you remember, when I say class switching, I said IG. What? When I say which specific IG what? Which antibody? I said you have to remember when I say class switching. IgM. I said IgM. C's. Remember to say complement fixation, complement activation, and class switching. So that's what the 3 C's I was trying to tell you. And then we'll talk about the diversity and how we respond to that. So anyway, basic genetics. So you can see we talk of chromosomes, we talk of genes and alleles. So each human cell should have 46 matched into 23 pairs. So we get 23 from the father and 23 from the mother, and they get together in our cell to establish what genes we have. And then again, we have roughly about 20,000 to 25,000 genes. So it says 25, good enough. So the segments of DNA that contain the instructions for making specific body proteins, so these are called genes. So these are the segments of that DNA, the double helix that you normally see over here as well. So these segments over here, these segments, shorter or larger, they will instruct the cell to make a protein. Which protein are we talking about? But remember, when I say that, today I'm going to talk about immunoglobulins, which are antibodies, and they are proteins. When I say receptors, most of the receptors are proteins. So that pretty much. We talk about alleles. So you can see that one of two or more versions of this gene. For example, if your father has a blue eye and your mother has a brown eye, so what do you get? So you get half and half from both. So it's going to be like inheritance, dominant, recessive, things of that nature, Mendelian, the concept that you have. So likewise goes into antibody structure. And you can see from here, these are in the nucleus, and there are different types of them, and 22 of them, and this is the one that determines whether you are male or female, X or Y. Okay? So these are for real, and they are there, and this is how they'll look like. And if everything is normal, it's normal, but the message of making proteins is there. And human genome has been mapped. That's what I was telling you. The new drugs are going to go and hit your genes. That's what the whole idea is. Human genome has been mapped a couple of years ago. So they have mapped what exactly needs to go, what actually is there. Now they are mapping other things as well. in terms of knowing out what's going on into the cell. And again, all of these things are in nucleus of each and every cell. Now, the other things that you may want to keep in mind is that when we talk of DNA, this is how the DNA looks like. When we talk of RNA, is a half slice of that. And then there's a process where, for example, DNA is replicated. And this is how the structure of a DNA is. And we will talk about cytokines, antibodies, and so on and so forth, and how the DNA is transcribed into RNA, and there's a process, and then RNA is translated into a protein. So there are sequences over here in terms of production of any protein. So these are like basic genetics, regardless of antibody, that I'm going to discuss today. And you can see from here, all these processes have regulators, they have enzymes, and there are a lot of cut and paste that takes place. And that's what we do in real time as well in terms of modern medicine. Again, you can see from here 1920 to 50, when actually Watson and Crick DNA double helix. And then again in 60s, you can see from here DNA code was discovered. That's why there is AGGC. And then again, the gene basically means a sequence over here, a small segment over here that bears different amino acids. And so that's gene for example, gene for making antibody, gene for making insulin, gene, you name it. So that's actually a portion of a DNA that will, of course, carry those nucleic acids, that you can see deoxyribonucleic acids. And then again, precise amino acid, and we have a sequence, and we can pretty much determine that. And that gives body a choice that we are all proteins, but proteins in the hair, in the nail, in the eyes, in the heart. They're all proteins, but they're all different. Okay, so that's where we need to understand in terms of diversity of antibody production. Now let's go to the antibody. So that was like basic genetics. And then you can see from here, so far, we've been talking about heavy chain and the light chain over here, right? And then in heavy chain, we talked about a portion which is constant. We talked about C portion. And I also told you that the C portion has, within that constant, we had this GAMD, gamma, alpha, micro, delta, and epsilon. So that's like a sequence over there. Then I also told you that you can see from here variable portions. So light chain, heavy chain has a variable portion. And I also give an example of you put on different gloves on your fingers, right? So these are like, if these are the sequence of amino acids on both sides, sequence of amino acid with your fingerprints as uniquely different. All of us may have 5 fingers, but all of us will have a different fingerprint. Exactly the same case in happens to production. Now, if you go to configuration, so there are different segments. Everything is coming from the top. So we have a bigger, it's like a pyramid. From a bigger choice, it's just like 26 alphabets and you're gonna make a word. So you, very rarely, you can come up with one single word that will have 26 alphabets. But you can try your best. So you have to pick and make a legitimate or a functional word that will make sense. So likewise, in production of proteins, you can see a lot of processes are happening. And if you look at the color codes, just like a basic structure, so you will see that there is a germline, there is a choice for you, and there are constant region exons. So introns, exons, exon the external portion, introns the internal portion of a gene, and codon. And what you will see is that the constant portions pretty much are stable as different. So who makes that choice? So just pick and draw. Who makes the choice? That you make a choice of all those 26, kind of alphabet. You make a choice with your finger, tuck, 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 tuck. You pick up, right? So in our system, we have enzymes, right? So these enzymes which recombine or recombinases. We call them recombinases. Simple word, A's enzyme. Cut and paste, cut and paste. Control V, control. So whatever it is. So we call recombinases. I'm going to talk about that. I just want you to remember in this class at least two recombinases, which we'll call RAG1 and RAG2. RAG1 and RAG2. And even if you miss some of the points, I think I'm pretty much going to repeat what I delivered last year. So you can go back to the lecture as well and help yourself, and I'm going to post that as well. But you will see over here, not much of a choice in terms of a constant region, right? But when it comes to the variable regions, that's why it's called variable. So now you will see a choice there. Okay? I think I should give an example. She said that, well, you would rather wear same shape and color and size of the shoes as compared to different type of gloves. So the constant portion pretty much is the same. That's why it's called constant as compared to the variable portion, which is in the hands. And you can see from here, so there are three major segments I want you to remember. They call VDJ. VDJ. And these are segments, and there are a fixed number of choices in our germline that our bodies had to pick up those segments. You have a choice. Okay? And you can see from here, when these choices are made, right, we call this, for example, the very first choice will be the D and J will be recombined. So we can pick up some of the J's or D's over here and J's. So that's the very first step in this case for antibody production. So we kind of pick and choose from this one. So you can see there are like 6, 7 of them. We just pick 5, 3. So we put them together, right? And we didn't go for a change for V segment. Then the second step is VDJ recombination. So you can see it takes place in a proper way. So by the end of the day, we have VDJ combination to appear as an assembly line where you have a constant, pretty much the same. You had a choice because you want to shrink the D and J segments and then the V segments. It's just like giving an example of, you know, when I say my office number is 773-821, right? 2165. So the chances are very rarely people will have the same number, right? But again, 773 maybe is area wise. So there's some familiarities. So likewise, there's a constant region for that part, which happens over here as compared to anything else. But I just want to convey to you that this is done, and I've put it very simply, but it's more complicated than what it looks over here because they are very specific enzyme. They are regulators, they are controllers, but also keep in mind things can go wrong. Things can go out of control as well. Okay, so what we are talking about is the structure. Now, again, the same thing. This is a typical, as you can see, prototype of gene that codes for transmembrane protein. Because remember, all of these are different proteins. So they are all different proteins. Each and every will have a gene, and each and every will have the regulator. And you can see from here, amino terminal and carboxy terminal. So this is a transmembrane region, and these are all these domains which are there. Now again, the same thing happens is in the genomic DNA, you have a 5' and 3' basic structure of a DNA, and then you have a gene that's firstly transcribed and then maybe spliced before it is translated into protein. So this is like basic biochemistry, that when you see difference between transcription, translation, and then mature messenger RNA. But keep that in mind, the body does it for itself. We also do that. So we have this ability to go into the protein and slice the gene, insert the gene. That's what biotechnology is about. That's what we normally do. So that's what the advanced genetics is, that we can put the gene. That's what we call sometimes For example, if a person has a genetic disorder, we find out this gene is missing. So we'll splice this gene from somebody else, and we have a gene gun, we'll put it there. So we kind of play around with stitching because we have enzymes. So that's advanced genetics and advanced cell biology, where we have the ability to do that. But before we do that, we want to know what it is. So you can see from here, there are steps involved at the cell surface where all these things are done before you get the proper structure of a protein. In this case, you can see, and there are different parts of this transcript. So these are different parts of transcripts which are coming from the genomic DNA, like exon 1, exon 2, exon 3, and 4. And then also remember, initially, these exons are joined together with introns, so they kind of put them together. But as you can see, the very first step is that you have a change of the introns, and then they are joined together by a transcript. And then again, these are also removed, so they are sequenced. So step by step, you have these introns, putting them together, joining them together, and they come very close to each other in terms before this is translated into the nucleus to start making those particular proteins in your endoplasmic reticulum, and then assembled and all that things that we've been talking about. Okay, so so you can see there's an organization element and there's a rearrangement. So that's what you saw. In the previous slide, I told you the rearrangement takes place for the light chains, because there was not much of a rearrangement for the heavy chains, except for the distant part. So that's another thing. And then also keep in mind that everything is regulated. If it comes out of control, it will be dysregulated, and that's what is the basis of? What is the basis? Say it again? Pathophysiology, cancer. Cancer, when everything goes out of control. So you start making too many proteins with no control. So this means mass expanding, and you don't have any control. Now in this case, another example of, if you remember, I told you two end, a amino end and a carboxy end. And then again, they have a variable portion and they have a constant portion, right? But if you go back over here, you see these variable and constant portion are coming from genes. So the very first thing, as you know, I have got my keyboard, everything is scattered. Then I say we would call like rearranged. So there's a Q, W, E, R, T. It doesn't make any sense though, but I have to kind of arrange that. First step for genes is that you have a choice, that you have about, in the variable region, you have choice of picking up at least like 40 different genes over here, if it comes from the variable. For joining, you may have 1 to 5. This is a fixed number. So you're going to pick and choose. So this is where we have the germline DNA, where everything is rearranged, and you can see each one of them carries a number, and each one of them carries a name. But by the end, if I was to slice that up, you know, so when I slice that up, so there is a head and tail, right? And there is, in genetics, we say 5 end and then 3 end. So if you notice, or we say carboxy end and the amino end. So you will see over here, that's why we want to suture them afterwards, join them. So you can say 5 end and 3 end, and then see this over here. So the very first thing that happens is, as I said, VDJ recombination. And I told you that that happens because of an enzyme recombinase or recombinases. And I wanted you to remember at least 2, which are regulatory enzymes, which are called RAG1 and RAG2. And some of the people will be missing that. That will be the basis of the problem, or basis of pathophysiology. So the rearrangement happens, especially for the B cell DNA. And you can see we pick up V1 and V2. We pick up J4 and J5, and these are introns. And we just pick up one of the constant portions. And it's in the constant portion. I think I did told you especially that there are lighter chains as well, kappa or lambda. I'm going to go in a little bit detail today, kappa or lambda, as compared to those 5 ones. And then transcription, further splicing of these things joined together is a mature missing RNA, and then translation, and then you got kappa chain polypeptide. So we have this kappa chain polypeptide. This is taken from here, put onto the tips of variable. So the idea is the distal end of those antibodies, they are specific because of all these steps. So this means whatever initiates the response in terms of the antigen is going to see an antigen and give you a very specific number that belongs to you only. So that is happening from the pool. So they have a bigger pool over here, and it may look simple to you, but even for the kappa chain over here has 40 different choices, and there are 5 different choices. And this case, since it is a kappa one, so constant region should not change. That's why it's called constant. So constant will remain. You can see through and through because it is a kappa chain. And then the variable portion over here may have a variable protein or amino acid sequence. Okay. The other thing that can happen, this is how the body does it. Because you see the linear gene. So you saw the gene segment, a linear segment, just for understanding. And it has a 5 end and a 3 end, and it's a germline DNA. And then you can see from here, we have this enzyme, I told you, recombinase. So that's the one who's going to make these choices. So recombinase will come and then recombine the recognition sequence. So that's what it does. So what it will do is that, for example, it wants to eliminate V3, V4, J1, J2. It's going to come over here, join these 2 portions, and cut it. And that's what we do as well. That's actually also the basis of the testing that we do for a gene, we call gene slicing. And we have the ability and we have the enzymes to do that. And that's, again, all the basis of biotechnology. And then again, you can see from here, VDJ recombinase, and then we can rearrange it and we can get it go. So we have a choice where we involved an enzyme, which in this case is recombinase, that is specific to make sure that we cut and paste, right? And this is something we call germline DNA. I'm going to explain that later as well when I teach you bacteriology and we talk of genomic DNA. Okay. Now, again, let's talk of, in this case, let's talk of, for example, a micro or gamma or delta, any of those heavy chains. So you again will see that they have a amino terminal, a carboxy terminal, right? And this one has a variable portion and a constant portion, variable portion and constant portion, right? So what is happening is, again, the whole idea for all these figures in your book is that you are seeing gene rearrangement and then subsequent steps and synthesis of heavy chains. And then you can see that body has this capability, we call it splicing or slicing in terms of the gene. Again, if you go on the germline DNA, we have a variable portion of the heavy chain. We're talking about the heavy chain, because heavy chain is a variable portion as well, if you remember that, right? Heavy chain is a variable portion, the inner leg. In this, we have a choice of about 50. In the D, we have choice of 20, and we have a choice of 6 in terms of J, and then we have a choice of constant gene regions. So you can see that's where we have all those GAMD things coming up. But I also told you that especially for IgG may have, I mean, initially we said only one, but it has more than one constant. So we have IgG1, IgG2, IgG3, and that's what it's coming from, because the reason is that we have gamma 1, gamma 2, gamma 3, gamma 4. Initially, when we thought it was 1, but we went down and researched, we found out no. But that choice is not for E so far. That choice is not for D so far, right? It may come up, but as it stands, the current book doesn't talk of that. So we know for sure, at the most, we have 4 different IgGs, and rest of them, other than A also, they think 2. But other than that, we don't have much choices in terms of constant regions, right? Again, we have RAG1, RAG2. So you can see that we are trying to now pick and choose for the constant region. So we come over here, we put a micro, for example. Let me take an example of micro, splice it up, and then add micro over here. And then again, as we talked about, the DJ combination first, and then VDJ combination, that's where the variable portion will come. So we will end up making different choices in terms of the final molecule that we may get. Okay, but what I'm trying to tell you is that now when I say IgG or IgG1, so you will just think IgG1 in terms of constant segment. But in terms of variable, we can have tons of choices. So this is a choices which is in terms of millions and billions. It doesn't seem like that. Okay, so even if you add, I mean, I just told you 26 alphabets, and there is no shortage of written literature different from other. That's why we even test you for plagiarism. You can't even copy and paste. So you have to. So every 5 billion people in the world are saying something, writing something. All is different, and the base is only 26 alphabet, and it will keep on. They never come up with this idea. Oh, let's increase that. No need to do that, right? Isn't it? So you can see from here, if that 26 can fill the libraries of the world and information overflow, you just have 50, 20, and 6, so like 76. Too much. So keep this in mind, the variability in terms of very specific antibody. For example, you'll ask this question to the Department of Health, hey, we may get this swine flu, why don't you make a vaccine for? So, well, which swine flu you want me to make vaccine for? The question is, the one that happened 10 years ago? Well, that may not come again. So the viruses will mutate. They will come up with new sequence of antigen. Unless and until we see that antigen, we cannot make an antibody prospectively. That make sense? At the best, they can make a what do you call a flu shot. The flu shot is for the virus that erupted last year, hoping it may have some cross-reactivity, similarity. It may give you, and they think maybe give you like 50% or more protection. Does that make sense? So likewise, we have very, very specific antibody titer in terms of, that's what we normally do. Okay? So far so good. I know it's going to get complex, but again, you really have to listen to it over and over again, read it over. But I'm not asking for much. I'm just asking you to understand the constant portion, a little bit of variability in the constant portion, especially with alpha 1 to alpha 4, and the variable portion of the heavy chain and light chain, and why is it variable, and where is this diversity coming from, and how are we able to generate 10 to the 11 different. So it is good for us that we can respond to millions and billions of antigen. Do you want to respond? No, we don't want to respond. That's another thing. Because if you start responding, you're going to kill yourself. So that's another concept we'll discuss later. But we have an ability to do that. We are designed for that, naturally. Okay? So let's go back now to other concept, which is class or isotype switching. Now, again, the sequence, and this pretty much is the same if you notice. This is a standard protein synthesis that you can read in biochemistry, cell biology, so on and so forth. There's always a DNA, there's always a gene, and there's a message over there, and the steps over there are pretty much, the first step is that transcription of whatever is required from the genomic DNA, and then translation, and then synthesis of protein. The standard, and there are tons of enzymes though. I'm not going to go in detail. I just will hold you responsible for just only one, or maybe two. There's another coming up, recombinases. Okay, now let's see, for example, and keep that in mind while I'm teaching you antibody diversity, but I also mean diversity in TCR at the same time. The reason is, if you remember, because both of them come from super Ig family. Both of them will have heavy chain and light chains. Both of them will have variability. Both of them have ability to recognize antigen. Okay, I'm just using this, your book is using that, but keep that in mind that the T cell will also be unique. As we have an ability to make a unique antibody to a unique antigen, so do we to make a unique T cell for a unique antigen. Sometimes vaccines are T cell based, some vaccines are antibody based. That's important. Now, let's go back, and you can see from here the class switch. Okay, the class switch, what it means is that we talked about B cell being mother, and I said she's going to give birth to antibodies, and then two of the antibodies, M and D, are going to attach on top of that as B cell receptor. And then I also told you that the very first antibody to appear is IgM. I also told you that IgM is first to appear. It looks like boss around, but we want a professional antibody. So we basically are looking for IgG, because it has very specific even 4, right? Now, antigen, when it comes in contact, right, with a cell, for example, right? Now, that triggers a response. That's why when you get infected, so this means that your B cell will see this antigen, they will start making IgM. But I just told you 2 P's for poor antiviral, poor bacterial. So why you want to have poor IgM in terms of functional? We would rather. But how is it the body is going to switch to IgG? So the ability of the body to switch from production of one antibody to a production of another antibody is called class switching, as simple as that. And that is controlled. So that is controlled. So your body is happy, everything's okay, and then they are making whatever serum half-lives of those antibodies are, everything's going well, all of a sudden you got flu. And as an antigen comes into the system, no, we don't want, we want to address that, right? So what will happen is that the antigen will come in contact with the T cell cytokines, which we'll discuss later, and we did discuss gamma interferon as a classical cytokine. So what will happen in this case is, now these are the C regions. So you can see all those regions are there, gamma, alpha, delta, epsilon. You know, they all are there in the constant region, right? Now at this step, what is happening is that initially I said the antibody was for micro, right? So the very first antibody the body will see is going to send the best person. Okay, we just got antigen, just kind of overwhelm it or immobilize it. So it's like jump on top of the antigen and just immobilize the antigen. Good enough for IgM to do that because she has 10 valencies, she's powerful, blah, blah, blah. But that's not the end of the problem. We would rather have IgG to take care of in a cleaner way. So what will happen is that now we want the class switch, right? So you can see from here that we want to switch from this one, which is micro, to gamma. So if this happens to be a series of that, which sends a message, make this. So we want to take that message out. We're going to rewrite the sequence. Take this message out, and we want to replace that, and we want to switch to, let's say, gamma 1, for example. So you can see we get gamma over here. So this step over here, which will now start, rest of it is fine, because you have a variable region, then you have specific to that antigen, and then in constant region, you start making gamma 1, right? Before you were making, for example, micro. So micro doesn't have 1, 2, 3, 4, it's just 1. So this process where we switch to gamma 1 and all the steps till we get gamma 1 polypeptide, and which was like a heavy chain, with of course variable chain, which is going to be antigen specific. The whole sequence that we call over here will be termed as class switching. And then again, we want to do that. We want to do that. And if you read your book, it says that the fetus starts producing IgM, right? And just for 5 months, and it stops. But if you were to find IgM after birth in the baby, so the question is whether it is of fetal origin or not, because baby is not ready for class switching at that time. She has just come into the world, right? So then you start thinking that if she gets IgM, which is not of fetal origin, so IgM is coming from the mother. But I also told you that IgM cannot pass through. So all these things are like relevant clinical questions, and you want to go in that direction. But just keep in mind that this is how they assess the genetic diseases. This is how they look at the patient and so on and so forth, based upon the primary information you get. Okay? So class switching. Now, again, you will see this particular slide is basically talking about some of the somatic gene conversion. Because remember, we get specific or diversity in antibody production by recombining the gene. One step is recombining the gene. So that's what we normally do, right? And sometime what happened is the second step is rearrangement. The 2 R's here, right? So kind of recombine or rearrange. So you can take, for example, a M micro and combine it with different one, or you can rearrange the same sequence. So 2 R's there, that's why we have recombinases. And I told you RAG1, RAG2. Now in this case, also keep in mind that we have this ability in our body, which we call it somatic gene conversion. We call this somatic gene conversion. And this basically phenomena exists in us, but this particular, slide doesn't talk of human. It basically is talking about the chicken immunoglobulins. With many of the experiments, and you wonder, why do we talk of chicken? Why do we talk of chicken immunoglobulins? Any guess? Why do you want to talk of chicken immunoglobulins? Say that?
Similar to human?
Oh my goodness. No. Chicken, no. Okay, let me answer. I don't want you to think too much. Yeah.
Much less variability?
Same thing. Pretty much he's the same, much as the variability. I mean, you are right. The only thing that we are discussing about that is how do we make vaccines for antibodies. So what we do is that we inoculate chicken eggs with an antigen, right? And that eggs produce antibodies. And these antibodies are purified. So we are interested in finding out what's happening in chicken. Okay? So we are interested for vaccine formation purposes. So if we were to make a vaccine, we need billions of eggs, especially chicken. That make sense? For some of the antibody production, for example, we don't want to give it to a human being and take his antibodies because it's going to be an ethical problem. So we would rather give it to to a horse or a bigger animal that has a lot of blood, then purify that and make sure that we cut and paste and make sure that it is similar. Because if it's not similar, then we will have transfusion reactions. We have a lot of react problems over there. So we want to make sure that it's purified. And there are many different ways. It's not that simple as I'm telling you, but I'm just giving an example. That this in chicken IG, but the process is the same. Again, you can imagine all these things are happening in animal response to an antigen as well. That's why we have bird flu and all these kind of diseases. Again, you will see there are some functional gene, and then in chicken sometimes they have pseudogenes. So pseudogenes basically get inserted into the functional gene. The idea is that we pick up an animal model and learn from there. And it may happen, it's all developmental stage. It's not the final word. Even the textbook that you're reading is 5 years old. So what Dr. Koiko and Sunshine have done is, last year they sat and picked out the research paper for the last 5 years and wrote up a textbook. So by the time, that's why we're talking about basics. So if you Google it and find out from PubMed, things are changing drastically. But again, for us at this level, we at least know there's something called pseudogene as well. Remember when we pathophysiology we talk of proto-oncogenes and oncogenes, so everything should make sense to us. What is there in terms of the genes being combined with the normal genes, and you may have a different product. Okay, now to summarize what I've gone through, and again, as I said, that you can read in the book as well. I've just picked up the salient points. Let's say variability in the H-heavy gene, and the reason is that since this is going to act as a BCR, which is a B cell receptor, we have ability to recognize different antigen. And this variability can be present, as I said, VDJ. So VDJ, V stands for variable, D stands for diversity, J stands for joining. So this was all those choices that you have, pick and choose from V, D, J, and constant. Okay? And then again, you can see from here the DNA from every cell in the body, which is the germline, contains multiple VDJ gene segments for heavy and light chain synthesis. So we have choices. Each of our cells contain that information. So we have the choices to kind of cut and paste into different levels. Okay? Now, we know for sure that once everything is assembled, so then it will follow the protocol. So this means that we will end up having 2 heavy chains, 2 light chains. We'll have constant portion and we have variable portion. Then the size and pretty much everything will be same, but the distal edges of the sequence of amino acids, the numbers, as I said, give you example of the fingers or fingerprints, that will be different. So that's how the whole synthesis happens. When we talked about class switching in the previous slide, again, normally it does not happen. It only happens when there's a need for it to happen, and that happens when there's an antigenic stimulation. So in antigenic stimulation, and you saw the previous slide, the heavy chain DNA has to rearrange. And then again, you can see from here, micro and delta because remember, why do I say micro and delta? Because remember, these were the 2 attached to the B cell, so they are normally produced, right? And so there are plenty of them. Now we want to switch to other ones, like for example, instead of normally producing IgM and IgD, we want to switch to IgG, IgA, or IgE, but with the same antigen specificity. So this is important. So this means once we pick up a variable portion, then that gloves, for example, we make those gloves for those antibodies. So whether it's say IgG, IgA, or IgE, they can put on the same gloves to deal with the same antigen. but their original structure will remain. So that's called antigen specificity. Though it's another story, and you wonder why do we want to go for IgE? If your body starts making IgE, so this will lead to what's very special for IgE? Allergy. So then we are going in another direction. Okay? And again, I just picked up salient point. It is beautifully described in your book, and once you have a base, you will enjoy reading it. When I said diversity, we already talked about that. So I tell you that there are variable regions in both light and heavy chain. Why do you want to have variable regions for both light and heavy chain? Why do you want to have that? Why do you have to have variable portion for both heavy and light chain? Because the Fab, the antigen binding segment has both. So if you say variability should be in both. So if you look at that in heavy and light chain, we have to have that. And then again, we talked about rearrangement and then assortment of HL, and then again, all the other things are important in terms of mutation. So I don't want to use this term right now, but the fact is that they were doing what they were doing, and all of a sudden they changed because of provocation by an antigen. So in genetic terms, we will call that mutation, but for a good cause. Normally you say mutation, you think of bad things like viruses mutating, but in this case, you want mutation would rather mean change, but as long as it's controlled, good enough. Okay? The idea is, why do you have these mechanisms there? These mechanisms are there because we can now generate a vast number of antibodies to different specific antigen specificities. I gave you an example of the bird flu. Because viruses have that's why we cannot stockpile anything for viruses, because they keep on changing. So there's always a big alert, something breaks out. And when I'll teach you virology, we'll discuss about that's the biggest threat to humanity, which has killed people in millions within a short span of time. Because of mutation, nothing will work. But again, we have ability to do that. And to summarize, you can see that why do we have this generational antibody diversity with all these things in there. And I just summarized it, and we already discussed it as we went along this different picture. And the last slide is, I told you that I wanted you to know at least two basic enzyme recombinases, RAG1 and RAG2. And I said you need to know at least other important enzyme, which is called activation-induced cytidine deaminase. It's called AID, activation-induced cytidine deaminase. And as the word suggests, you can see there are a lot of players over here. The most important thing is activation. And this is the major enzyme in the class switch recombination. So all those events that are there, we talked about, like recombinases, this will get a signal from the body, hey, we need to change. So what is it that initiates that? Right? That's what when we say there's a signal in the body, we say, hey, start making, stop. So there's an initiator or stop for everything. So for example, your regeneration of hair is taking place because signal says start, stop, start, stop. If it's out of control, it's only start, no stop, then we have a problem. So in this case, the most important thing is activation-induced cytidine deaminase, very important for class switch recombination, very important for somatic hypermutation, and also important for gene conversion. So these are the three major events which are happening in this case, especially for the ability of antibodies to be very specific and very diversified in terms of response that we normally get. Okay?